Hi, this is Sahana here with gene interaction. So to begin with, Mendel postulated three laws based on his experiments, right? So the first law is law of dominance. So this particular law states that in heterozygous condition, which appears in F1 generation, okay? So in heterozygous condition, the allele whose characters are expressed over the other allele is called the dominant allele. So the character that expresses itself is called the dominant allele, and the character that gets suppressed by the dominant allele is called the recessive allele. So the dominant allele produce their character. So that particular character is called as dominant character, which appears in F1 generation irrespective of the presence of recessive allele, whereas the recessive allele which gets suppressed, whose character gets suppressed, is called recessive character, and recessive characters always appear in F2 generation. So these are the examples of dominant allele, and these are the recessive allele. So that's what law of dominance states. One of the allele is dominant over the other allele in heterozygous condition. Now coming to the law of segregation. So this particular law states that each individual has two alleles for a particular character, be it homozygous dominant, heterozygous, or homozygous recessive. So each character is governed by two alleles, right? So at the time of gamete formation, the alleles separate in such a way that one of the allele goes to one gamete and the other goes to the other gamete. They are not mixing up at the time of gamete formation. Each of the alleles going to a different gametes here. So this is what law of segregation states. So this particular law is called as law of purity of gametes, because the gametes are always pure. They either has any one of the allele. Fine. Now, third law posed by Mendel is law of independent assortment. So this can be seen in dihybrid cross, where assortment of one gene occurs independently of other gene. Okay, so this other gene is a non-allelic one. So the segregation or the assortment of this particular gene is independent of this gene. So that is what independent assortment. So these two R and R are allelic genes, and these are the non-allelic to the R. So when you consider only R, they are, this capital R and small r are allelic. This capital Y and small y are allelic. When I consider capital R and capital Y, or vice versa. Okay, so these are the non-allelic ones. So this is a different group of gene, and this is a different group of gene governing two set of characters. So here, at the time of gamete formation in dihybrid cross, when this actually assorts into a gamete, this is not dependent on the other gene pair that is present. So randomly this goes to one gamete, and this the other gene or non-allelic genes goes to the gamete. So this is what independent assortment states. But there are few concepts that does not hold true for this particular independent assortment, right? There is linkage and gene interaction. What actually happens in linkage is, say for example, there is these two are the gene pairs governing two particular character. So this is for a round, and or this is for a flower color red, and this is for a flower color, or flower color or pod color yellow. So this particular character at the time of gamete formation is going to the one gamete, whereas in case of linkage, there is a chance that the non-allelic gene can also link to this and go to a particular gamete. So that's a whole new different concept there. So that is what happens at the time of gamete formation. There is a chance that these gametes, that can be allelic, non-allelic, or from the same loci, or from homologous chromosomes, or from non-homologous chromosome, the gene anywhere from the chromosome can affect the character or trait of one particular gene. So which means that is not actually following the independent assortment rule. So this actually can be called as deviation from Mendelian ratios. So we'll be dealing gene interaction and linkage separately in coming classes. So this particular concepts are not come along the independent assortment law of Mendel. So this can be called as deviation from Mendelian genetics, or the monohybrid and phenotypic and genotypic ratios will be different in this particular thing. So we can also call it as modification of Mendelian ratios. Coming to the gene interaction or interaction of genes. So this is the expression of a single character by the interaction of more than one pair of genes is called gene interaction or interaction of genes. I told you, right? So here one particular character is governed by more than one pair of genes. Fine. So for example, if I take the tall plant, so the tallness of the plant is governed by the capital T, small t, tallness and dwarfness. But to uh, to alter this or to modify this, there is some other gene present in different loci, or maybe different chromosome, or maybe a different locus of the same chromosome. So there are different genes that is actually having a influence on this particular gene that is actually deciding the height of the plant. So that we'll be discussing later. So in interaction of genes, there are two major types. One is intragenic, or it's also called as allelic, and the other one is intergenic, which is non-allelic. So, coming to the intragenic, in science, the simple thing is just go through the name, understand the name or the title. That explains most of the concept. See, intragenic, genic is the genes, intra is the within the genes. Okay, the interaction happening within the genes. Okay, so what is there within the genes? There are alleles. So within the gene, interaction is happening and the gene has alleles. So this can also be called as allelic gene interaction. Okay. So the definition goes like this: the genetic interaction between the two alleles of a single locus or single gene is known as allelic gene interaction. This can also be called as interallelic interaction. Interallelic, why is it is happening between two alleles, two contrasting pair of alleles. So that's why it can also be called as interallelic interaction. So there are different forms of allelic interaction that is happening. One is complete dominance, where the character or the trait of this particular recessive allele is masked by the dominant allele. So this can be a allelic interaction where a character is influenced by the other allele here. So incomplete dominance is there, codominance, polygenes, pleiotropism. So these particular types will be discussing individually in further classes. So the next one is intergenic or non-allelic gene interaction. So this particular interaction is the interaction that occurs between two independent genes. Okay, this is the important word here: independent genes located in different locus of the same chromosome or different chromosome. Okay, it can be these two genes which are non-allelic. Non-allelic, why? We know here R and R are allelic and R and Y are non-allelic here. So the character of this particular allele pairs or gene pairs are influenced by the other genes here, other alleles here. So this particular alleles can be present in the same chromosome but in different loci, or it is there in the same chromosome but in the different arm, or it can be in totally different chromosome that is influencing on this. It can be anyhow. Okay, but the thing is here here the one pair of the gene is getting influenced by the other gene which is non-allelic to this particular gene. Here there are different forms again. There is epistasis, supplementary factors, complementary factors, and so on. But these are the important forms here. So as I said, we'll be discussing these all these types and forms individually in the for the coming classes. So till then,